Welcome, Chameleon Academy, to Season 7 of the Chameleon Academy Podcast. We are here at our first episode starting this new season, 2022, and we are starting the year strong. Today, I'm going to bring you an interview with Jonathan Hill of I, Pardalis and Benson Morrill of Rare Genetics, Inc., and a project that they're putting together. It's now being funded on GoFundMe. It's going to allow you to send in a sample of shed skin from your Panther Chameleon, and they're going to be able to tell you what locale that Panther Chameleon came from. And so if you're wondering if your Panther Chameleon was really ambilobe or really ambanja, then now you're going to be able to find out. Of course, this raises other questions about how disruptive this may be to what we think we know, but that's all part of moving forward. Now, that's enough of me talking here. Let's go ahead and bring on the experts to explain the program in their own words. And so please join me in welcoming Jonathan Hill and Benson Morrill to the Chameleon Academy Podcast. Welcome, Chameleon Wranglers, to this episode of the Chameleon Academy. I'm your host, Bill Strand, and today we're going to be talking about a very interesting genetic test that a couple of members of the community are thinking about doing on Panther Chameleons. And so I'm going to be introducing you to Ben Morrill of Rare Genetics, Inc. and Jonathan Hill of I, Pardalis. Welcome, gentlemen, to the podcast.
Thanks for having us, Bill.
Now, I'm going to start with Jonathan, since you're the originator of this project. Could you explain what this project is and what you hope to accomplish by sponsoring this project?
Sure. So I guess I'll start with how did we come up with this idea? I've been using Ben's services with some of my snake projects for gender tests because it's a non-intrusive way to figure out gender. And so every time I submit my tests, I always think, man, it'd be great if we had a test for panthers, which is primarily what I do. And so the last time that I emailed Ben talking about the gender test, I asked him if this was in development, if a Panther Locale test had been considered before. And so it just kind of naturally came from that I'd used the gender test in the past and had always wondered, could we start a project for Panther locales? So now it was just a follow-on question.
Now you're talking about gender, but now you're switching to locales.
Yep. So big switch.
This is, yeah. So what is exactly you are trying to determine with this project?
So I've always, as anybody who's worked with Panther Chameleons knows, locale is a big part of the hobby. Most breeders, one of the first questions they're going to be asking themselves is, do I want to work with crosses or do I want to try to stay pure to the locale? And many breeders would prefer to stick to one locale, but generally the way that we determine locale isn't very scientific. It's what the importer said first. That starts the chain in the pet trade, and then second, what we know about the locale, like wild pictures of animals in specific locations, previous imports, and just what people expect in the captive breeding community. And so sometimes those are the same thing, sometimes those are different. Like Nosy B, everybody's kind of come to expect a true blue when in the wild that's not what you see 100% of the time. And so it's interesting that the way the hobby has developed, locale's central to it, but we've never had really kind of an objective way to check it or to understand it. And it's always been this trade knowledge or expertise that breeders develop and importers are familiar with, and then everyone else trusts. And so that's kind of that's central to what we're trying to help with. But then there's these specific things like Florida wild caughts. There's the Florida population of panthers, and there's a lot of rumors that recent wild caughts are just Florida passed off as true Madagascar
Mm.
imports. And there's also people who have gotten a female in an import and they aren't sure if she's properly labeled. There's a lot of these specific challenges that breeders face that I believe a test like this would help us solve. And not entirely, but it would help us provide some more information in that whatever situation you find yourself in. So.
So, with this project that you're doing, and let's go ahead and describe the project. You've got a Kickstarter right now. You're starting.
Yep.
What is this project? What are the inputs and what are the results, the outputs of
Sure.
this black box
Sure.
project?
So I asked Ben for a quote and then put together a budget of what I thought was reasonable for the project and created a GoFundMe page for it to start collecting money because I also thought this doesn't it doesn't really make sense for one person to sponsor this. This is something the whole community is going to benefit from. So it has a natural kind of it should be funded by everybody because we're all going to benefit kind of um, position, and so that led me to think, man, we should just GoFundMe. We did create a GoFundMe for this. What's going to be required for the test? A lot of Ben's tests they're using shed skin from reptiles, so it's a certain quantity of skin that's required to run the test. The base data that we have so far, we're going to be doing a lit review. That's part of the beginning is lit review and research and development. So the initial idea that we have could change, and what's possible is also going to be determined when we develop the test. So that's where we are. We're collecting funds. We have an initial idea and initial requirements. Eventually, we think it probably be within the range of $75 per test, but it could be a little more, a little less, depending on the volume that comes through and how it's developed. But the when we were initially starting some of that lit review and building the GoFundMe information, we found the data that was submitted with the publication of Gerbic et al. I'm not sure how to say his last name, but GRBIC. And they did a really good job collecting wild caught specimens, geocoding the specific location where they got the blood sample. They took pictures of those animals, and then they published the DNA, the pictures, and the locations of 324 wild specimens. So when thinking about a project like this, data is usually the hardest part, like to even get started. And so by finding that, I got really excited. I was like, wow, this is great. That's a pretty decent sized sample. 324 is a big number for a species like Panther Chameleons. So pretty promising to start out.
Okay, so say this project goes forward. At the end of the project, you're setting up a system where anybody in the community could send in, say, shed skin or whatever sample you determine, and
Yep.
they could get a response back saying this is what locale or what combination of locales is within your Panther?
We wanted the combination. I think the more we look at it with the haplogroups, it's either going to be a match or not. Not much of a percentage, but we'll see. It depends. I think initially, though, it looks like the mitochondrial DNA haplogroups have really like pretty good separation when you look at different locations in Madagascar. And so every location that they sampled, if you're talking about like Ambelobe East, Ambelobe West, a number of mutations that separate those two populations is a minimum of 6. Some are up in the range of 40 mutations. So that's really good. And what that is it's a unique set of mitochondrial mutations that you don't see anywhere else. You only see in that sample in that location. And those mutations take a long time to occur because mitochondrial DNA is inherited from the mother. And usually I did a lot of research trying to figure out how long does it take for those mutations to occur, and different species, different reproductive rates, etc. And I've come up with like a ballpark couple hundred generations is when you would expect a single mutation. And when you see 6 mutations of separation, that means those two populations have been isolated for a long time.
Okay. Let's go over to Ben. Please give us your background, and then let's start digging into the science behind this and all those biological terms that Jonathan just threw out at us that
Sorry.
not all of us may totally understand. So let's start off with your background and what your part is in this.
So as far back as I can remember, I've been catching snakes and lizards and, whatever I can find. People have asked me many times, when I started liking snakes and all the animals normal people think are icky, and it's just always been that way as far back as I can remember. Probably very similar to many people listening to this.
Oh, yeah.
So I've always had that love. When I was in high school, I called the local university. I grew up in northern Utah, and so Utah State University was the closest to where I lived. It was just a 10-minute drive. I called, and I think I was a junior in high school, and I just called the biology department and said, "Hey, is there a herpetologist there that I could talk to? I would love to learn more and be able to come and see a college lab." And thankfully, there was a really cool professor there. His name is Joe Mendelson, and he's now the curator of reptiles at the Atlanta Zoo. So I worked with him. He was, like I said, really patient with me as a high school student. He let me come into the lab and meet the grad students. And a year or two later, I helped with a stomach contents study where we had buckets and buckets of road-killed snakes from Arizona and New Mexico, kind of on the border, southern part of those states. And so we were keying out the goo from inside of these snakes. And then I've always really liked molecular biology as well. That ended up being more, I worked in Joe's lab for a while, and then there's another professor, Mike Frender, and I think where's he at now? I can't remember now, but anyway, he was another person early on that helped me quite a bit, and his lab, they were much more focused on molecular biology. And so starting as an undergrad, I started doing both reptile stuff and molecular biology. I got to do a little bit of field work. I got to work at the field station where all those road-killed snakes came from, Portal, Arizona. And that was a lot of fun. But I didn't get to do very much. I was mostly in the lab, so lots of molecular biology background. And so to kind of fast forward through, I got my undergrad in biology, and then my PhD is in animal science with a emphasis in molecular biology. And my PhD ended up being on the quantitative genetic analysis of reproductive traits in ball pythons. So I was able to study Dan and Colette Sutherland were in California, and then they moved to Utah. And when they moved to Utah, I started talking with them, and Colette had, stacks of handwritten notes. She had measured the length, width, and weight of individual eggs. And I think it was something like 10,000 eggs or something over 10 years. And so I was able to do some molecular biology to be able to show genetically they had produced some twins. They were identical twins, and they did have one set of twins that were fraternal. And so that ended up being my PhD. And then I moved into my full-time job. I now do molecular biology work and embryo work with pigs to be able to make their genomes, specifically their immune system, more human-like so that their organs can be used in transplant in humans.
Oh, very interesting. So what is the science behind what we're trying to do here in establishing locales and being able to identify locales?
So the biggest thing, first of all, when John first reached out to me, it's a tough thing to develop a test for. And one of the first things I told him is, we have to have samples from individuals we know where they came from. Otherwise, we don't have a way to compare. We can't say it's from this locale. We can say that this person believes it's from there, or, something like that.
Yep.
I was like, I'm going to need to have at least a couple dozen samples from places where that it is correctly identified where it came from. And so this paper that John found, they did an amazing job being able to have over 300 DNA samples from blood draws. That's awesome.
Yep.
And all of the pictures and all of that, I don't understand that part of it, but the DNA side of it, that was awesome.
Yeah.
Okay.
So cool to see that. And so that's what really springboard that, went from one of the many emails. I'm answering people's questions and thinking of things, and there's always cool things that pop up that people are interested in. But this one quickly jumped, close to the top of my list because we have that resource. So what they're able to do in this paper is they're using the mitochondrial DNA that John was talking about earlier, and then also a spot in the genome, the nuclear DNA. So you have, when you're talking specifically about DNA in an animal or plant, you have two different sources. In plants, it's the chloroplast and then the nucleus. In animals, it's the mitochondria and the nucleus. Yep. And so the biggest difference is the nuclear DNA you're going to have in lizards and in humans and many animals. Plants, it's a little bit different sometimes, but in animals, you're going to have two different copies. One came from the mom, one came from the dad. And then in the mitochondrial DNA, it's only from the mom's side. And you don't get any information about who her dad was. All you know is the mom, and then you know about her mom and the mom before that and the mom before that. You don't get any information about the sire's side of things. And so it's kind of two different things that we can look at, and what John was talking about earlier, where it's really unlikely to have a change. If you remember in like high school biology class, when you talked about crossing over and segregation, so in mitochondrial DNA, none of that happens. There's only one copy, and it just comes from the mom. There's no segregating. There's no crossing over. There's none of that. Those mechanisms that are in place in the nuclear DNA. It's important to have that in the nuclear DNA because of changing environments. You want to be able to have a genome where there's genetic variation, where you have different things that you can do. So the mitochondrial DNA is different. It's staying the same, much more likely to be unchanged, whereas the nuclear DNA is more likely to have changes. And then when you look at the nuclear DNA, there are different levels that you can see there as well. There's parts of the DNA that are vital for a cell to live. And so those DNA sequences don't change much at all as far as the nuclear DNA goes. So the nuclear DNA that they're looking at in this paper is a part of the genome that doesn't, the nuclear genome that, as far as talking about nuclear, doesn't change very much. Yep. And then some other places we can look, kind of the complete opposite side of that. So like if I wanted to tell from a DNA, like maybe say it's a poop sample in the wild, and I wanted to know if it's a chameleon, it's a snake, it's a rat, it's a bird, I would want to look at one of these spots in the genome that doesn't change very much, and I can actually use the same primers, and I can get 16S is a really common one, and I can just use one set of primers and I can amplify a bit of that DNA from lots of different animals with that one primer set. And then looking at what the sequence says, I can just blast it. I can do a search in online database and it'll tell me what species that came from. On the complete opposite end of that is if I want to be able to do paternity testing or tell the difference between two individuals or that are closely related, then I want to look at things like SNPs and microsatellites. So SNPs are single nucleotide polymorphisms. So it's a single, like in the sequence, it's a T and then it changes to a C. Mm-hmm. So some individuals will have a T, some will have a C. And so if I look at my dad's DNA, he might be heterozygous for a specific base where he has a T and a C. Well, half of his kids, I might have a T, my sister might have a C, we'll get half and half, and then whatever we get from the mom's side. So it's a way to be able to tell a really fine scale. So it's like zooming in and looking really close at small differences because those parts of the genome are not expressed, usually aren't making proteins. They're not important for life. And so specifically what these researchers have done with the Panther chameleons was they used a really, really, zoomed out look at the very general DNA in the mitochondrion, and then also a part of the DNA that's much more general in the nuclear DNA. But even though they're looking at the very general parts of the genome and of the mitochondrion, they're still able to see significant differences in 11 different, they show in the paper 11 different groups. Mm-hmm. And so what I suggested that we could do is we can start having some samples come in once we have some money to be able to, do the literature review, order primers and get set up. I have to test things. When you've done this enough times, that sometimes it doesn't work just like it is in the paper, or you have a different machine or something like that. So it'll take a little bit of time to get up and running. But once that's going, then samples can come in and we can see, okay, this is a sample that's unknown. This is a sample we think is this. This is a sample that we think is that. And once we can actually run them, then we can say, okay, this one matches mitochondrial and both copies of the nuclear. So we kind of will have three different pieces of information. We'll have the mitochondrial, then we'll have the mom's nuclear signal, and then we'll have the dad's nuclear signal. So we'll have three different things that we can compare. So if all three of those match up to one of those locales or, their groups that they have, then, that's a pretty convincing match.
Yep.
If two of the three match and one is something else, then we have to see, okay, is that one that's nearby? Maybe it was a natural, something that could have happened in the wild, but from their paper, my understanding of it, it seems like they have pretty significant segregation, separation of those locales, which is pretty amazing.
Yeah, it depends.
So that's what we'd have right off the bat.
Yep.
And then when you talk about percentages, so like if you want to test some clothing from a crime scene or a hair follicle from a piece of hair from a crime scene, and that comes through and they say, okay, it doesn't match anybody, but we know this person's Caucasian, that they blah, blah, blah. Like they know that's coming from snips and microsatellites. And so for me to be able to say, okay, I know this one's a mix, we want to be able to say it's what percent this, that, and the other. We would need to do some more work to be able to develop, to really zoom in and look at the really small differences in between individuals. And for that, it would be awesome if we could get the DNA from those 324 and just develop the microsats on them, be able to see what they have, but that's probably not possible.
Yeah, that was one of the things.
If we can come up with something like that, then that would get us to the point where we can say the percent, or kind of like the dog, if you have a puppy and you want to know what percent Golden Lab and what percent, this and that, you're looking at snips and microsatellites for that.
How exact is this science? As in, when you get these samples in and you have something to compare to, is there going to be a lot of a human interpretation, or is it just yes, no?
The nice thing is, if it's a perfect match to one of the groups, then, that's easy. I can just say, okay, this matches perfectly. If I see something new, or if I see something where, there is a base change that matches one group, but there's also in that same piece of DNA, there's a change that matches another one, but that exact combination didn't match any of the 324 that they looked at, then, we have to kind of just do the best we can from that bit of DNA that there's, something different going on. From what they showed in that paper, it seems like the majority of the time we'll get those right off the bat of those three pieces of information, we'll get an answer that allele came from that group, that one came from that group, or all three came from this group. But yeah, it won't surprise me at all if we get some new things that pop up, new combinations that weren't sampled in that, and that's possible. And we just do the best. I would say that the best way to think about it is it's going to be a heck of a lot better than what we have right now. Might not be perfect, but
yep.
How much coordination do you need to do with the scientists that did this paper that you're basing this on?
The really cool thing about DNA sequencing is they just put it all out there. Obviously some things, like if it's how a drug affects gene expression and cancer cells, obviously they're not going to publish that. They're not going to make that available. But any of you, just like John did, you could actually go to NCBI. That's our United States National Center for Bioinformatics Information or something like that. But NCBI, you go on there, you type in Panther Chameleon sequence, you go to there, they have this drop down, go to nucleotide, and it'll just bring up everything that's that species and DNA that's been published. So all those 324 samples, they've put that all up there. So I can go anytime, I can pull all those sequences in, I can throw ours in and compare it to it, see how it goes. Another thing to your question earlier, if we do get something that's different from those 324, we do have really nice software that's been used for many years, phylogenetic software, where I can have those 324 samples in there. And that's how they make these trees, these cladograms, to be able to tell which individuals are closer related to others and things like that. So I can take a new sample, a new unknown, and I can throw it into that software and it'll put it in the tree where it belongs. So it wouldn't be me guessing, okay, well, maybe that T, blah, blah, blah. Like I can just put it in there, and all the sequences that they've made publicly available, anyone can access. Anyone that learns how to use that software, they could do it themselves. They get their sequence information from us, and they can just plug it right in and see where it lands in that tree.
Okay. And have you looked at the information enough to be able to answer the question, how different are the locales at that level?
The wild ones in that paper, yes, but I would love to see at least that. Like I told John, I would love to start with a group of 25 samples from people. It would be great if it's a mix of ones that they feel pretty confident that this is pure this or that, and then other ones that they're pretty confident that it's not pure anything. Yeah, I'd like to see about a dozen of each. Because right now I can't tell you anything about the captive population. And you all probably know better than me. If I went out and got, let's say I go to Daytona Reptile Show and buy a Panther Chameleon, how many generations back do you think it was in the wild? How likely is it that individual is wild? How likely is it it's been captive bred for 10 generations? I don't know the answer to that. You guys might.
If you're at a Florida show, it all depends on which booth you're buying it from.
How about Chicago?
Yeah, Chicago's a little bit easier, but still they'll ship them up there. But it
really depends on whether it's a breeder or not.
Yeah. Bill, I after downloading the data, I opened it up. I looked at the Python code because I'm a data scientist by trade, so I use Python every day, and looked at the data, looked at the haplogroups, and wrote an article with my understanding of what is being said in the paper. And one of the interesting things is on the East Coast, so towards more of the population centers on the East Coast, like Andapa, Simbava, and Talaha, those areas, those three, for example, are in one haplogroup. So what we would consider three locales has one haplogroup. And what that means to me is we consider them in the pet trade locales, but they interbreed enough that when you look at their genetics, there's not as much separation. And there's a couple of examples like that where the haplogroups, where you're actually mapping out the genetic distance, where those don't line up with what we call locales. So, and another really interesting one is Ambilobe East and Ambilobe West. In the captive bred trade, we just say Ambilobe, but there's a haplogroup in the east that's at a high elevation. And as you run down the road towards Darena, I think is how you say it, but as you run down that road at a higher elevation to the east of Ambilobe, that population is different, genetically different from the population to the west as you go down into the coastal plains towards Sirama. So when you read this paper, it's really cool because we treat those as one locale in the trade, but there's evidence that they really are two. And there's probably there's probably tons of examples like that in Mbanja with the sub locales, but they didn't sample those micro locations as well. They just sampled Mbanja generally. And there's one haplogroup over there that's it's a sample, and they were like, there's something going on here. And they, as researchers, didn't really know, but I looked up the lat lons of that one that they couldn't really explain the latitude and longitude of that one, and it's in Ankaphi. So for us, we're all like, oh yeah, it's Ankaphi. That's why it's different. and so like it's actually a lot of fun to get into that paper because there's certain things that if you've been in the trade breeding panthers for a while, you know what locales we would expect. There's some surprises. There's also things that make a ton of sense. So,
okay. So to get this all done, let's go back to Jonathan. You've started a GoFundMe. At this point, what needs to be done for this to be a go?
So we just need to get to our budget so that then we can get going on the research and development. We created tiers in the GoFundMe to go get those samples from the community. So there's a tier 1, tier 2, and then platinum. And each tier you can submit more samples into the R&D. But really we need those samples. So we're combining the two needs into one thing there. So once we get those 25 samples that Ben was talking about and we hit the budget, then we'll get started.
And so this is, as I understand, this is set up so there isn't an expiration date. It's just as soon as it's live until the budget is hit, and once the budget is hit, it's a go.
Yeah, and if I get impatient, then I might just fill the rest. So.
All right, Ben. Say Jonathan calls you up, says, "Hey, we hit the budget." What happens then?
Then I say, cool, I will get back to you probably in 2 weeks after I've done a really deep literature review. I'll look up snips and I'll look up microsats. I will type in the genus and species, and I will look at every single sequence that's been done, regardless of where it is in the genome and what it's from. So I'll know everything that's been done, specifically amplifying DNA, especially from wild samples. I won't care so much about captive ones. So yeah, that part will take me a couple weeks to really deep dive in and know what's going on, what's been done. And then during that time, hopefully people are collecting samples, and anyone listening now, I strongly encourage you. The cool thing about the sheds is you want to remove it from the cage as soon as you can, especially if it's a humid cage. I don't know how humid. Seems like chameleons, you keep them pretty
humid.
There's a pretty good amount of airflow, but there is still some humid spots. Yeah. So anyway, you want to the enemy to good DNA is moisture. And so as soon as you can remove it from the cage, what I usually do is I have a Ziploc baggie already labeled for that individual, and I have it sitting either on top or to the side of the cage. And this is especially helpful if you're collecting a bunch, which I'm always collecting from, a bunch. So I have that bag sitting there. When that shed comes, I just pull that shed out, I set it on top of the bag, and then I set, a little toy or something on there to keep it from blowing away or anything. Let that completely dry out. And once that's completely dry, put it in the bag, and that DNA will be good even at room temperature for years. I have some that have been in my basement for 4 or 5 years. It's kind of damp down there, but since they're in Ziploc bags, they were dried and then they're in Ziploc bags, they still work. I still get good DNA. I can still amplify off of them. So start collecting sheds now, and then that will help us. As soon as the money's there, I'm looking at the literature and ordering primers. That's the other thing I'll do is I'll order the primers they had in this study for sure. And then any other snips or microsatellites or other things people have done, I might order some of those as well to try them out. So we want to have sheds coming in during that time.
SMP.
Snips and microsatellites, what are those?
So the SNPs are the single nucleotide polymorphisms. And so that's where randomly in the genome. One, let's say both of your
So
parents,
helpful. Thank you.
both of your parents have a T, but you end up having a C there. That would be a new SNP, and that's a very rare event. That's like, 1 in millions kind of a thing. So it's just where one base changes. And so it's a really nice, when you want to really zoom in and look at small differences between individuals, we'll go to a part of the genome that's not important for life. So it doesn't express, it doesn't make a protein, it doesn't, have anything to do with the cell living or dying. And so parts of the genome like that, you're able to have changes occur in those spots, and that offspring still lives because it didn't, if you're looking at something like 16S, that's your ribosomal DNA that, then leads to your ribosomes. Your cells can't live without ribosomes. So if there's a DNA change there that makes a stop codon, so that protein's not made,
fatal,
then that fetus is aborted.
Yep.
That, never lives to see the light of day. And so we look in this kind of junk part of the DNA. We used to always call it junk DNA when I was in school in 2000. We didn't understand that some of that intronic sequence does get used for some things, but those base changes can be there. And so that's why we want to look for those SNPs, is because they're small changes that do occur a lot more frequently than a change in the sequences that are important for cellular life, and everything that would lead to a baby being born and being alive. And the microsatellites are very similar. It's just a bigger change. And so what we'll do is we'll go in, like in 2002, the first time I did any microsat stuff, we were looking in pine beetles for designing new microsatellites. And essentially what we did is we did a bunch of sequencing, and then we were able to kind of purify to get where we have these tetranucleotide repeats. So it'll be like a TTAA, TTAA, TTAA. There'll be like 18 in a row where it's TTAA, 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 TTAA. And because it's so repetitive, when the polymerase goes to copy that to make the next, if that cell's splitting, if it's a sperm cell or an egg, since it's so repetitive, sometimes the polymerase makes mistakes. It's a lot more likely to make a mistake when it's repetitive like that than if it's just A, T, C, G, A. If it's just, a nice changing, then it won't make that mistake. But if you've got all this repeat going on, it'll mess up. And so that's another place in the genome where if you want to zoom in and tell differences between individuals, those microsatellites are really helpful.
Okay. So Jonathan calls you up and says, we're a go. And then there's 2 weeks of you gathering all this information. At the end of the 2 weeks, are you then ready to receive your shed skin?
Yeah,
yeah.
And
when somebody when someone sends in their shed skin, how long after that until they get an answer as to the locale match?
It will depend on. What I would like to have is a look at the first set of 25 before I give anybody any information, because I really kind of need to get that global view of, okay, yeah, this is really cool. All of these seem to fit, like those 3 pieces of information all fit one of these groups always. Or, oh, geez, this isn't very helpful at all. We don't see what they're seeing in the wild. And this test is complete junk. I mean, that's possible. From what they showed in the paper, I think it's extremely unlikely, especially because this is a part of the genome that does not change very much.
Yep.
Like Jonathan was saying, your chances of getting a change within 200 generations is your chance is only 1 out of 200 generations. So I would say
to get more than 6,
I would feel uncomfortable giving much information until we've gone through. So I would say that's kind of your beta testing. If when the 2 weeks are up, I've already got 25 sheds, I can run them all in one run, then we have an answer, in a couple of weeks after that. I would sequence it, do the data analysis, kind of talk over it with John and other people that are, an active part of it and have good. I obviously don't have any background knowledge of these localities and what they look like and things like that. So that will be a very cool, fun part to take the data from those 25 and be able to talk about, what we're learning and what's fits and what doesn't. And that's when I'd happily write something up that we could, have some pictures. You guys add some pictures in, and I can put some, of these phylograms, trees, and show where things are fitting. And I think it'd be pretty fun.
Okay. So how would somebody send in a sample? Say after the first 25, you're going to be collecting the first 25, but after that, once this is up and running, is there going to be something on Rare Genetics Inc.?
Yeah, so the way we're set up to accept samples is really cool, really easy, really nice. We're very fortunate that we're able to just get a shed skin, completely non-invasive way to collect a sample. With other species like birds, they have to, a lot of the time they need to go in and see their vet, or they have to pull a feather, deep enough that they're getting some of the root and send that in. So we're really lucky. So you would go on our website, like right now, the most common test we have is for sex determination in colubrid snakes. We have a really good test that works across there's like 3,000 different species of colubrids, and it seems to work on all of them so far. So what they do is the person would go online, they pay $15 for that test. It's a really simple, easy test, much simpler because there's no sequencing there. So they go on, they pay the money, and then they get an order number. And so all they need to do is take their shed that's dried and in the bag and put it in the envelope, and then have that order number written. They can print out their receipt or just handwrite it on there, the order number. And when it comes to us, we know who it is. We get their email address, phone number, way to be able to give their information back once we have results.
What does it take to be able to set up a test like this to tell the gender of, say, baby Parsons chameleons or adult Mellors chameleons? My audience is going to ask that question, so I may as well get it out of the way.
And that's something I would love to look more into. My first question, just because I don't know chameleons very well, is, are they temperature dependent sex determination or genetic sex determination?
We believe they're genetic.
So then it's possible. The first species that are temperature, I cannot help you.
Okay.
So as long as it is genetic sex determination, there is an actual DNA difference that leads to a male or a female, then we can develop a test. And I'm not familiar. I can't tell you right away what the sex chromosomes are like. Some species have, like humans, the X and the Y, you have this giant X and this little teeny Y. There's lots of differences. So it's really easy for someone like me to develop a test. I can tell X apart from a Y. Yep. But in pythons and boas and snakes, the X and Y are identical. Even like the different stains they do, different things, like you can't really tell a difference. And still to this day, I have gone, the chromosome that they're on is 65 million bases, and I have it down to about 1 million. I know it's somewhere in that million. Okay. And this year, I'm pretty sure we have some people we're collaborating with that have a genomics center that are going to help us with the bioinformatics side of it. We'll be able to do that. So even if it was the Parsons chameleon, you said?
Yep. Yeah, as a baby,
and so even if, even if their chromosomes, yeah. Now, even if their sex chromosomes are really similar, with this help from this Genomics Institute, we should be able to figure that out pretty fast.
Right. And Bill, by the way,
sorry.
Yeah.
No, I was just going to say, Bill, that's one of the cool things about this. As talking to people after putting it out there, I've heard so many ideas similar to that, where they're like, "Oh man, it'd be great if we had a test for Mellors or Parsons." Xanths versus Hawaii imports on the Jackson side. I mean, there's a ton of things that'd be pretty cool to be able to just test for.
Yeah. Now,
and the biggest thing that helps make it possible now is sequencing costs are way different. Like when I was in college in 2000, that human genome sequencing project cost millions of dollars and took several years. And now, within the next year or two, you should be able to do an okay whole genome sequence of a human in like 2 or 3 days, and it costs $100 as long as you're doing 200 or 300 at a time. You can get that cost way down, and so that's why talking to me today versus even 5 years ago, I wouldn't be as excited and positive about it. But now the sequencing costs have come down so much that I can take, 10 known male parsons, 10 known female parsons, and I can just sequence their whole genome. And then the bioinformatics side is so good now, they can plug it into their pipeline to do because you're looking at millions, billions of bases, A's, T's, C's, and G's, but they have software figured out to where it will be able to help us figure out how to tell those two sex chromosomes apart. So it's good now.
And by the way, just as a note, I have to say that I have always heard that the sex determination was genetic in chameleons, but now I'm hearing from some breeders that they are seeing some differences in temperature, and so it's murky at this point, and so don't take what I said as the gold standard. There's still
I'll look it up.
Yeah, so,
I'll look it up and see if they have done karyotyping and talk about sex chromosomes. Then I know that's a good sign that at least those researchers thought it was genetic sex determination enough to come up with this, karyotype where they're showing sex chromosomes, and that would give me something to work from.
So once you have this established, how much is the test going to be?
So for the locality test, where we're sequencing 3 different parts of the genome, that's much more complicated than the sex determination test I was talking about. So I would hope it's going to be right around $75, give or take a little bit.
All right. And this question is for Jonathan. What do you think this is going to do to our community? Go ahead. Spitball.
Yeah, I know. So I've gotten a lot of feedback, both positive and some concerned, like people that are concerned about potential drama related to test results.
No.
And I can relate to that concern. I mean, because if you spend a little time on social media, then it can get ugly. But the way I see it is some of that is just a function of social media, and actually all of it. Because if any of us sat in front of each other, we wouldn't say any of this stuff.
There's already drama on there right now with no test.
Yeah, I know. And so that's the first point. The second I'd say is I could see a lot of beneficial use cases for this almost immediately, as a breeder, but also as a hobbyist who's just curious to know more about their animal. I think that I've heard some people concerned about if we determine that there's a lot more crosses out there than we thought, that will devalue certain animals or things like that. I don't think so. I mean, I think at the end of the day, people love panther chameleons because they're beautiful, because they're amazing animals, and we are primarily interested in making sure they're healthy and enjoying, like, watching them thrive in captivity. So for people who are all about the money, yeah, maybe for them, it's going to devalue their product. But that's not me. That's not most of the people I like to work with. And so I'm less concerned about that, than maybe others who are all about the money, but like to me, the way I see it is, I just want to understand my breeders. I want to understand their genetics. And I find it really interesting. So, I think it could be, beneficial for a lot of different things. So,
and I think that it'll be interesting to see what we find out and how much of our ambulobes or umbanjas are actually crosses, and then I would imagine that it's just going to elevate the color to a name. Say this is a yellow-bodied, blue-barred panther chameleon. If we find out that the yellow bodies end up being a whole mix of all sorts of different stuff, just like the blue panther chameleon. Yeah, it's from Nosy Bay, but it's really not a Nosy Bay. It's just a subset of what was found in Nosy Bay. So it's a blue panther chameleon. So maybe we'll have, like crested geckos, they all have the different colors that you can get. We'll see.
And as someone who works with ambulobe, when I saw the east-west divide and the high elevation in the west versus the low coastal plains in the east, and seeing that there's a genetic test that can tell me, hey, this animal has genetics from the higher elevation point, then I'm going to be even more interested in things like cold nighttime drops, and other things for that animal specifically, because we've always had this question of like, should every locale have the same husbandry? And we don't even know what the locale is. So, but what's really interesting about that is if we could say this is Ambilobe West, we can then have a decent basis for the husbandry decisions we're making. So I get excited about that. I think that's really cool. And
I
and
agree,
for
'cause-
so yeah.
Yeah, I mean, Panther chameleons, they're found from 0 feet above sea level to 1,000 meters. And so, we've always asked, should we be treating these differently? And this would finally give us the indication as to which ones we should try and see if we can find some subtle differences that are not noticed. Yeah, okay, they live, but maybe they live better. And we can start unraveling that. So this is an exciting chapter in our community.
Yeah. So
all right.
I understand. I understand the concerns, but that's my perspective. Is I just get excited about it. So.
Okay, so we're excited about doing this. What do we have to do? Where do we go to make this happen?
So there's I could share the link to the GoFundMe. We're getting close.
Yes.
We're getting real close to the budget.
link will be on the show notes to this episode, and I imagine you have other places, iPardalis.
Yep, iPardalis.com. I've been publishing some blog posts there. Ben's going to publish the results once we get to the next step. So those are all good places to go.
Okay. And so, everybody, if you know where Jonathan's website is, iPardalis, you can go there or go to chameleonacademy.com and find the show notes to this episode. And you'll be able to find the link there. And we're going to, I'm sure this is going to happen because I know there are a number of people that will make sure it happens and make sure it hits its goal. And I will be coming back around sometime later this year. And let's see what comes out of all of this. I'm excited. Jonathan, Ben, this has been a wonderful conversation. Is there anything else do you think we should add before we sign off?
I told John he needs to write a book once we do this.
Okay.
Yeah, and then I told him that I have 2 young kids, a full-time job, and a side business. So it's squeezing a book in there is going to be tough.
So it's a maybe.
Yeah, it's a maybe.
All right, I'm going to come back around with you guys a little later this year, and we are going to keep tabs as to what's going on. I want to thank you both very much for coming on, and we'll see what comes of this.
Thanks, Bill. Really appreciate
Yes,
it.
thank you.
Bye. Thank you, Jonathan. Thank you, Ben, for coming on and explaining your project. I think this is an important thing for us to do. If we can put together a test that identifies locales, we should do it. Now, this is going to raise an important question in the community. What happens if we find out that the things we called ambilobe or ambanja actually end up being crosses? And this can easily happen with exporters mislabeling females. So this may be a very eye-opening and awakening study because a number of us, me included, are saying that we should keep the locales pure. Ambilobe, ambanja, nocibe, don't mix them. And yes, some people are more militant than I am. I'm okay if you're gonna mix them as long as you tell somebody that you're mixing them. I don't recommend it because it's hard to sell a mix and they don't all come out a total bag of Skittles looking great. Some of them look more muddy than the originals, and so it's harder to sell them, and what happens to those? And especially the females. Mixed locale females are incredibly hard to sell. So from that aspect, I discourage it. From the aspect that once we mix locales, you can't unmix them. And there may come a time in the future where we aren't able to import panther chameleons anymore, and we're stuck with what we have. Once we get through mating all the different bloodlines, there may come a time where we are forced to mix the locales just to get the new bloodlines. And I would rather wait until that is thrust upon us than have it hastened upon us by us mixing things now. The more we mix, the sooner we're going to get to this homogeneous panther chameleon and lose all the differences. I'd rather that be far in the future. But what happens if once we get these tests, we realize that these very bright colors that we've come to accept, like, okay, the blue panther, that's the subset of the nosy bay. We know that. But what about these yellow-bodied blue bar ambulobes? They're wonderful. They're great. What if we find out that those are actually a mix of ambulobe and dapa or whatever? Does that mean that that animal is less valuable in the community? And I don't believe that. None of the animals that are here in the United States will ever go back to Madagascar. So the only reason why we are keeping the locales pure is so we can retain that individual beauty of each locale as long as possible. My personal feeling is that some of the color variations that we've created here, whether they are naturally occurring or not, are just as valid as the locales. Now keep in mind, that's my opinion. There's going to be a number of people that vehemently disagree with that. But if, say, the yellow-bodied blue bar ambulobe, just for an example, turns out to be a cross, I don't think that makes it less valuable. It's still a distinct animal. And if we no longer are able to call it an ambulobe, then we call it a yellow-bodied blue bar panther chameleon. It is a distinct color form. And me personally, I'm okay with that. So what will happen with the results of these tests? Simply, we'll have more information, we'll know the truth, and then we're just going to adjust to it. We'll at least be able to do away with people fighting over proving which locale they have. Now, we'll know. And as soon as this GoFundMe is funded, we will find out and we will see together what happens. I'll definitely be covering what's going on and I will report back here. So subscribe to this podcast on whatever podcast playing app you have or subscribe on YouTube and you'll get a weekly dose of Chameleon Academy Podcast. Now, the best way to stay current with the Chameleon Academy Podcast is to download the free app from the iTunes Store or the Google Play Store. This app will contain all 6 seasons and the 7th season of the Chameleon Academy Podcast, and you'll be able to reference any episode in the entire history of this show, as well as any new episodes that are launched. If you're on YouTube, subscribe to the Chameleon Academy YouTube channel, as I make educational videos and I put them there. Now, if you want a daily Chameleon touchpoint, go to the Instagram account for chameleon_academy, and you will get your fill of chameleon goodness there. Thank you very much for joining me here today, and I will see you next week.